Yes. Okay, great. Thank you. It's a pleasure to be here to represent Class 2. Today I'm going to talk to you about memory and the aging brain. And let me see if I can do this. Yes. One of the greatest scientific puzzles is how the brain captures and preserves the unique experiences of each individual. This organ gives us our own identity and the ability to understand other great mysteries that exist around us. As I reflect back on my career trajectory, it seems that I was really fortunate to have been part of a young field of neuroscience that was just getting its footing in the late 1960s and early 1970s, exactly around the time that I headed from California to go to graduate school in Canada, much to my parents' dismay. There were a number of fundamental discoveries being made in the field at that time in the areas of cellular, computational systems and cognitive neuroscience that I was fortunate enough to have been exposed to in graduate school. Just as a backdrop, it's been known since the 1950s that within the brain, the structure called the hippocampus plays a very special role in memory. After the neurosurgeon William Scoville performed bilateral removal of the hippocampus in a patient with severe epilepsy, the neuropsychologist Brenda Milner studied this individual, then known as HM, and discovered that he could no longer permanently store most information. Now, this focused really a spotlight on the hippocampus as a structure actively involved in laying down lasting memories, and questions began to arise concerning why damage to this structure had such a profound effect on memory. In the psychology department at Carleton University, a preprint of the monograph by O'Keefe and Nadell was being passed around among the graduate students, and this was the precursor of their full book, The Hippocampus as a Cognitive Map. Tolman originally developed the idea of a cognitive map as an organizer of cognitive operations in 1948. But O'Keefe and Nadell proposed that the hippocampus provided the neural basis of such cognitive mapping, providing a framework in which items and events could be represented in spatial and temporal context. In the early 1970s as well, the graduate students in our psychology seminar were also reading a monograph by David Marr. The hippocampus is a three-layer cortex, or archicortex, and Marr laid out a computational framework for how the anatomy of the principal cell types in the hippocampus determine its function to be a memorizer, much as in the same way that different cell types in the sixth-layer neocortex determine its functional role to classify. Emboldened by these rich ideas about the role that the hippocampus plays in memory, the fact that the hippocampus was structurally similar across all mammals, and the new perspective emphasized by O'Keefe and Nadell that the biological basis of human experience could be studied in animals other than humans, that seemed the right time to ask questions about how memories were laid down in the brain and what mechanisms might be responsible for age-related changes in memory. When attempting to determine what was known about memory and aging in the early 1970s, the views at that time were in line with some of the earliest written accounts of how memory changes with age. This quote, of course, comes from Aristotle, where he describes the very young and the very old and how they're defective in memory. And he said it was because they're in a state of flux, the former because of their growth, the latter owing to their decay. Just for background, the two most studied categories of cells, of course, in the brain are neurons and glia. Example cells are shown here. In humans, though, there are 86 billion neurons and 85 billion glia. Now, glia-neuron interactions are critical for brain function, but I'm going to focus just on neurons for this talk. So, when I went to the library to determine what was known about the brain and aging, these are the kinds of example cells these are the kinds of, alrighty. These are the kinds of example cells that I found from people across different ages. These are entorhinal cortical cells, and you see the progression of what was thought to happen to these neurons during the aging process. By the time you get to the example cell from a 90-year-old, it would be very shriveled. And of course, this is a particularly disturbing picture of brain aging because neurons are postmitotic. That is, they don't re-enter the cell cycle like skin or liver cells. So your neurons are born largely before your birth and die with you at the end of your life, with the exception of neurogenesis. So if this classic view of brain aging was correct, then no wonder the idea in the early 70s was that not that different from the view expressed by Aristotle. That is, if you live long enough, dementia was inevitable. Well, the reasons for this misconception include cell counting methods, but also, importantly, I think, an influence from stereotypic views of aging that have their roots in the social fabric of our society and not in scientific or experimental truth. Fast forwarding to more recent experiments employing increasingly rigorous methods, there is actually no cell loss of principal cells in the hippocampus, not in rats, mice, dogs, monkeys, or humans where they've been stereologically counted. Also, we know that now that many functional properties are preserved in hippocampal cells in the aged brain. We have found that membrane properties of hippocampal cells from old animals are similar, actually, in most respects to younger animal cells, and you can see the properties listed here. So, not only is there little cell loss in aging, but cell physiology is not dramatically altered. So we can make the inference that high levels of brain function is a normative part of aging. So what do I mean about healthy, normative, or typical aging? Well, from a brain perspective in humans, I mean those individuals without neurodegenerative disease that results in dementia. And the demographically most representative figures that we have for the United States are that 14% of people over 71 years of age have dementia. The most prevalent being, of course, Alzheimer's disease, the next vascular dementia. So today we know that becoming senescent or reaching a very old age does not imply neurodegenerative disease or that results in dementia. The other thing we know is that of all animals, humans spontaneously develop Alzheimer's disease, but other animals do not. So the prevalence of dementia, of course, in humans depends on the age range that you're looking at. Between 71 and 79 years, the incidence is 2.3% for Alzheimer's disease, whereas if you look over age 90, that goes up to 30%. So for this talk, I'm going to focus on what happens to the brain and cognition in the 86% of us over 71 who are aging normatively. One specific type of memory that the hippocampus is involved in is a spatial memory, the kind of memory that allows us to find our way through our environment, to know where the Kavli Auditorium is for now. So for humans, an example of how we know the hippocampus is critical for navigation and spatial memory is a positron emission tomography study, a PET study, that allows brain activity to be measured via glucose metabolism. Eleanor Maguire and her colleagues in London had taxi drivers recall a route while they were actually in the scanner and imagining that they were traveling with a passenger. And the right posterior hippocampus was powerfully activated, which you see at 3 different brain levels here in yellow. So for monkeys, we've also been able to show using FDG PET methods that when monkeys engage in behaviors involving navigation, such as walking back and forth on this 6-meter track, they also show the highest activity in the right posterior hippocampus. And this is true for both younger and older monkeys. For rats, John O'Keefe first shows that single cells in the hippocampus increase their firing rates when animals move through particular places within an environment. And this video shows the activity of one hippocampal cell. The black is the path of the rat in the box, and the red dots reflect where the cell is active. John called these cells place cells, and the area over which the cell fires as that cell's place field. So presumably then the firing pattern of many cells over a given space combined to form what O'Keefe and Nadel called a cognitive map. And these observations actually were critical to the development of the theory in their book. So I've shown you data from humans, monkeys, and rats that all support the idea that the hippocampus is fully engaged in this type of spatial navigation. Now, because there is no requirement for language to test spatial memory, it can be examined under similar conditions across species, and it can be examined across age. In fact, spatial memory has been shown to change at the age that is considered to be old in a given species. So this is 2 years for a rat, 22 years for a monkey, 65 years for humans. For rodents, an example spatial memory task is the circular platform. We developed this task in the mid-1970s for old rats, actually. And the object is to find the one place underneath this brightly lit platform and escape into it. And the consistent finding here between age groups was that older rats did not remember the location of the gold tunnel from day to day, as well as did the younger rats, suggesting a spatial memory deficit. In fact, not only rats, but older monkeys and older humans also show spatial memory impairments. And because of the spatial memory problems implicating hippocampal dysfunction exist at whatever age a species is considered to be old, it appears that there's a fundamental biological process that occurs in tissues of the body and also in the brain that is essentially sped up in rodents and in monkeys compared to humans. So from this, I think we can also make the inference that changes in memory are a normative part of the aging process. If in the hippocampus there's little cell loss and the basic physiological properties of these cells are preserved, why do we have spatial memory problems as we age? And of course, there are multiple answers to this question, and I'll discuss several. They all have to do with the synapse and the 100 trillion connections made by synapses onto neurons that are fundamental to network communication and ultimately for behavior. The first is that there's a change with age in numbers and functional state of synapses. And in aging, we know that depending on which hippocampal synapse you look at, there are either fewer actual synapses or fewer functional synaptic contacts. And another factor that contributes to memory change across age is altered mechanisms of plasticity at the synapse. And the famous Canadian psychologist Donald Hebb predicted in 1949 that the alteration of strength of these synaptic connections is the change in the brain that underlies memory. But it wasn't until 1973 when the Norwegian electrophysiologist Terje Lomo discovered a biological process that seemed to fit Hebb's postulate in the mammalian brain. And this biological process, of course, now is called LongTerm Potentiation, or LTP, and can be experimentally induced by delivering patterned electrical stimulation to axons that mimic normal neuron firing in the brain. When we studied LTP at hippocampal synapses in older rats, we found this type of plasticity to be altered. Under some conditions, LTP can be just as powerfully induced in young and older animals, but the decay of this modification in synaptic strength over days is about twice as fast in older rats compared with younger rats. And the change in this plasticity mechanism was correlated with changes in how good an animal's spatial memory is. So durable long-term potentiation at your hippocampal synapses was related or predictive of good memory in the circular platform spatial memory tasks, both in young and older animals. So clearly, the plasticity mechanism presumably used for information storage in the brain is compromised in old rats, and this appears to be correlated with individual differences in memory performance. The final change, the third change I'll mention, has to do with the fact that neurons do not function individually, but rather as networks to produce behavior. And I told you earlier about hippocampal place cells and how firing of multiple hippocampal cells are thought to contribute to and form a cognitive map. These two panels illustrate place cell recordings from a young rat on 2 different sessions in the same days. Different colors reflect different cells, firing patterns of place fields. And you see that the same hippocampal map is retrieved on the second session of the day as was there on the first. So the young rats retrieve the same map from session to session. And actually, most of the time, older rats also show accurate map retrieval. But about a third of the time, for any given old rat, a completely different place field distribution appears when the old rat goes back into the same and familiar room, as though the old rat had retrieved the wrong map. So this occasional failure of network stability is very likely to contribute to changes in spatial cognition with age. So in winding up here, I indicated in the beginning of my talk that there's been a fundamental shift in thinking about brain aging over the past several decades. The picture of normative aging that we have is very different than previous, in the 1970s. We know that memory does change with age, but we know now that not all of us are destined for Alzheimer's or vascular dementia. We know that we lose synaptic connections and functions with age, but we do not lose, there's no widespread neuron loss. Furthermore, even though there is synapse loss, there is also evidence for active adaptation in the aging brain in ways that suggest outright compensation. So, for example, those cells in the hippocampus that lose synapses, we discovered that the synapses that remain in old animals are stronger. So the cell can become active with fewer inputs. This may be evidence for biological compensation at the single synapse level. So we know, again, that there's reduced synaptic plasticity, but the synaptic modification can occur in the older animal. And we know that there are changes in network dynamics as we age. But again, there are experiments that clearly show that older individuals can recruit additional brain circuits to achieve memory retrieval that is equivalent in accuracy to that of younger individuals. So this may be then evidence for aging brain adaptation at a network level. Taken together, the older ideas that brain aging, that the brain ages passively is simply the wrong way to understand the changes that occur with age. Many of the insights that we have today about brain aging and cognition come from basic science work in rodents and non-human primates, as has been the focus of my laboratory. But today, we also need to begin to translate our knowledge of the aging brain and what factors impact cognitive trajectories into solutions for individuals. At the University of Arizona and in conjunction with the McKnight Brain Research Foundation, we're beginning to harness the power of precision medicine to predict health risks and personalized brain health interventions to maximize cognitive health span. This precision aging approach aims to close the gap that currently exists between optimal cognition and lifespan to increase the quality of life. So what can you all do here? Well, keep those synapses active by learning new things like you're doing today at these short talks. Thanks. And my acknowledgements for my former and current contributors to my thinking and experiments who have enriched my career, of course, my funding sources, and my family. Thanks.
Thank you, Karen.